Marchese Alfonso Cordi describes the cochlear receptor organ in the inner ear. He came from a noble family in Lombardy that resided in Sardinia. He studied medicine between 1841 and 1845 in Pavia. Bartolomeo Panizza showed him how to use a microscope, and he later learned methods to look at the membranous cochlea underneath it. After he made the discovery of the organ of Cordi, he got married and developed rheumatoid arthritis and fell silent from the scientific world for 22 years. Cordi was able to study the cochlea of 200 men and different animal species. He was introduced to the auditory system by family friend Antonio Scappa. He was the first to term sensory epithelium, the spinal ganglion, or spiral ganglion, the tectoral membrane, and the stria vascularis of the inner ear. He learned to prepare the microscope slides containing membranous cochlea using fluid or resin and staining them with carmine solutions. The cochlear nerve, afferent axons, are bipolar neurons, and their cell bodies lie in the cochlear nerve ganglion. They receive information from hair cells and project this information to the medulla. 95% of these axons are receiving information from the inner hair, and they use glutamate. The efferent cochlear nerve fibers come from the superior olivary complex in the medulla. These nerve fibers secrete acetylcholine. The organ of Corti is termed as the body's microphone. It sits in basilar membrane, and it contains 4 rows of cilia, and these move in response to the fluid pressure that creates an action potential. The perception of any loudness is connected here. Tiny movements of the membrane are enough to trigger an action potential, which will send a signal down the cochlear nerve and ends up in the auditory areas of the brain for processing. Cells work together to make the hearing possible. The hair cells is where the transduction happens, and the cilia sticks out from them. Cilia are surrounded by potassium cells in the fluid. If the ion channels open, which lets in the positively charged ions, the cell which would then depolarize and create an action potential. At rest, some potassium gets in through leak channels. There are more numerous hair cells, and they have the cilia inserted into the tectoral membrane. Insertional plaques hold the tip links to the cilium. The inner hair cells do not connect with the tectoral membrane, and 29% of those are inner cells. Thank you.